Hi, everybody, and good morning. Today, I would like to talk about transcranial focused ultrasound. And specifically, I would like to talk to you some of our work on the offline effects of this relatively novel technique. Now, luckily for me, there's a whole session dedicated to ultrasound later today at 2:45. And I realized that not everybody might be as familiar with this technique. So if you're interested, come this afternoon. But for now, there are a few points that I would like to highlight. So we're excited about this technique because potentially it allows us to stimulate with a superior spatial resolution and especially with a superior depth of stimulation compared to other non-invasive brain stimulation techniques. So here I'm actually showing some simulation work by a study performed by Legon, where they're stimulating in humans the thalamus with an incredible precision. It's a very active field of research on what the mechanisms of ultrasound are and how it modulates neuroactivity. But for now, I think it's important to mention that there is no evidence that at low intensities there's any effect of cavitation or thermal modulation, but actually that most evidence is here for a mechanical interaction on membranes, changing membrane potentials. If you've been following the literature, you might have seen very recently 2 papers coming out in Neuro. And I would like, I'm just showing one of the results from one paper because actually they're telling a very similar story. I'm taking a little bit of time here. And in this particular picture that I took from the Sato paper, they're doing calcium imaging in mice. And you can see here what the response of spiking activity will be evoked by a visual stimulus, by a flash of light. And in the middle panel, you can see that the visual cortex of the mouse lights up in one hemisphere only. This is the response to an auditory stimulus. And you can see the 2 auditory cortices lighting up in calcium spiking activity. What happens after an ultrasound stimulation to the visual cortex? I guess the authors might have expected that what they would see is an evoked potential in the visual cortex. That's what they stimulated, indicated by this dark red, black circle. Instead, they observed a response very similar to an auditory response. The interpretation that this is an auditory startle. But I would like to highlight, even though some people have interpreted this as ultrasound does not work and it's all just an auditory startle, that in fact in these studies the ultrasound was modulated or even in square pulse delivered that are well within the auditory range of rodents. And actually they're only testing for a spiking activity. Most of the evidence for ultrasound is a modulation of ongoing or evoked activity. And perhaps important for the studies I'm going to present here, I'll be looking at a different species, primates, namely macaque monkeys. And most critically, well, nearly all studies on ultrasound so far have focused on the online effects during the stimulation or immediately afterwards. We were really interested in the offline effects. We thought if there's any potential to develop this as a therapeutic tool and even as a research tool, it would be critical to show that the effects of ultrasound, the neuromodulatory effects, outlast the stimulation. So that's what we set out to be doing. And this is a project led by Jérôme Sallet. We delivered a train of ultrasound for 40 seconds, and 3 macaque monkeys, lightly anesthetized, went into the scanner about 40 minutes afterwards, and we scanned up to 2 hours later. So this is the period where we're going to look at their neuroactivation, and we're specifically going to look at their activity patterns. We were stimulating 2 brain areas to start off with. This is the supplementary motor area and the frontal pole. These areas are very distinct in the connectional profiles, and they're also far apart, so we can easily distinguish them, and we can also be sure that our relatively focal ultrasonic beam is able to separate them. Now, some safety aspects first. In collaboration with Jean-François Aubry in Paris, thermal stimulation here in the color panels depicted with the ultrasonic coupling cone at the top, and then in black outlines the skull, and below would be the brain, reveals that most of the acoustic pressure and most of the acoustic energy is absorbed by the skull, heating up to nearly 40 degrees, but actually the temperature rise after 40 seconds of stimulation, the train that we used, is a fraction of a degree. And to jump to the end of this study, histology did not show any change in microstructure. Now, to get to the meat of it, I'm going to show neural activity and functional connectivity of the macaque brain. I realize this is not part of everybody's daily work. So this is the macaque cortical surface. And on your left, you can see a medial view, and on your right, you can see a lateral view. And for everybody not accustomed to monkeys, luckily they're all primates and they're relatively similar in their organization. Occipital cortex is at the back of the brain, frontal pole at the front. Now, let's get started. And in a controlled condition without any stimulation, we're seeding functional connectivity in the supplementary motor area. And what we, the whole brain connectivity pattern we see validates that this is a sensory motor network. The question is, what happens after 40 seconds of ultrasound? Two effects were apparent. One, very close to SMA, the connectivity seed, we see that there's some evidence for enhanced coupling to regions that SMA is already strongly coupled with, while nearly all other regions in the brain, including the prefrontal cortex and the temporal lobe, have decreased coupling with SMA. SMA's connectivity profile is well known, and we can summarize this in what's called a connectional fingerprint. Let's follow the blue line here. This is SMA coupling strength with all of the regions on the circumference of the polar plot. And again, it's mostly the dorsal medial sensory motor network that SMA is coupled with. But if you compare this to the red line, after ultrasound over SMA, you can see these two effects clearly summarized. And as a control stimulation, this was the frontal pole, that in many respects is very similar to no stimulation. We were able to look at some of these temporal effects because we scanned for a longer time. And there's some evidence that the disruption of ultrasound on functional connectivity was apparent especially early on, but that there was a delayed appearance of the enhancement of local connections. And both of these effects seem to dissipate slightly later. But please note that this is very late. This is nearly 2 hours after the induction of the stimulation of ultrasound. In the last part, I would also like to highlight that we're able to stimulate deeper brains of the brain. And this is work led by Davide Folloni, who is interested if we can use a deep cortical region, the ACC, and a deep subcortical region, the amygdala. So let's start with the last one. At the top, you're going to see the control connectivity network without stimulation, and at the bottom, it's after ultrasound. And again, we see a very similar disruption of the connectivity profile of the amygdala. Here, summarized in a fingerprint, control, no stimulation is in blue, and the amygdala stimulation is in yellow. You can see that many areas that would otherwise be strongly connected with the amygdala are now disrupted. Let's go to the ACC. In a control condition here at the top, you can see it connects strongly with cingulate regions, with the prefrontal cortex and some parts of the default mode network. And all of these, again, are disrupted by ultrasound stimulation. And that's when you compare here the blue lines, no stimulation, to red ultrasound over ACC in the fingerprint. At the very end, I would like to give you some flavor of the behavioral impact of ultrasound. And this is work led by Elsa Fouragnan, again, together with Davide Folloni. And they were training monkeys who were interested in decision-making. And these monkeys had to pick between two options with a different probability of leading to a reward. And those two options were coming from a set of 3. So in each trial, a monkey was picking one object, not choosing another, but one object was unavailable. So there's one chosen object, and there would be 2 alternatives. Several brain regions are interested and are coding for the difference between chosen and alternative objects. But of those regions, only the ACC actually ranked the alternatives depending on their value, to a better value or a worse value. And of course, this ranking of alternatives is really important if you're actually going to have to choose between those alternatives at a next stage, at a later trial. That's exactly what the monkeys did, here depicted in this behavioral plot, in a control condition. Namely, they were more likely to pick the better alternative in upcoming trials. Now, specifically, this behavior was disrupted by ultrasound, a 40-second ultrasound train over ACC, a nearly chance difference between the better and the worse alternatives. Now, it's not that we were completely shutting down the brain or that the animal was not behaving in any sensible way. Namely, their performance here as a function of task difficulty was very preserved. It's actually a very specific modulation targeted at ACC and only interacting with the ACC processing. And actually, This, of course, also leads to differences in exploration and exploitation, as would be expected. If you're interested to learn more about modulations of self-connections by ultrasound or by the spread through a network, maybe you're interested in non-neuronal effects of ultrasound on the cortical spinal fluid or in the FBC that I didn't show here, and the validation that we ran in 3 different animals, or you're more interested in the ACC and decision-making, have a look at these preprints or come see our posters. But for now, I would like to thank you for your attention and all our collaborators.
Thank you very much. Is there any